So what I talked about in my presentation at ADPD was related to the fact that α-synuclein was initially discovered in the synapse and also in the nucleus of cells. But for a long time, researchers have focused primarily on its role in the synapse, not so much on what it might be doing in other subcellular compartments like the nuclei. So in our work, for a few years, we've been looking at the presence of synuclein in the nucleus, and we've reported this in mouse models, in cell models, and now we looked in human brain tissue. So we confirmed that α-synuclein does indeed occur in human brain tissue, and we found some changes, namely the phosphorylation on serine 129 that is considered a pathological hallmark of α-synuclein, and we found that this modification is increased in the brains of patients with dementia with Lewy bodies. So that's one important result I mentioned in my presentation. Then we also looked at what could be the consequences of the occurrence of α-synuclein in the nucleus. And in this context, what it could be doing is to interfere with gene expression. And gene expression is regulated at multiple levels, such as epigenetic level, epitranscriptomic. So I reported on some very recent findings where we find that α-synuclein overexpression in models of Parkinson's leads to epigenetic changes, so changes in DNA methylation, and also changes in modification in mRNA that affects its stability and its activity, and this is the epitranscriptomic angle of it. So these were the major messages of my presentation.